My name is Mark Linga, and on behalf of the MIT List Visual Arts Center, I'd like to thank you for coming out tonight for this public program presented in conjunction with Amalia Pica's exhibition. For those of you who haven't had a chance to see the show, I would really encourage you to come back this weekend as the exhibition closes on Sunday, April 7th. Amalia Pica was co-organized by the MCA Chicago, and after the show closes here, it's going to travel to Chicago where it opens to the public on April 27th. So this weekend is indeed the last weekend to see the exhibition, unless you all are planning a trip to Chicago at some point later this spring. The act of delivering and receiving verbal and nonverbal messages and the literal and metaphorical figure of the listener are central to Amalia Pica's work. And in conjunction with Pica's exhibition, the list is pleased to present tonight's talk by Sophia Roosth, Techniques of the Listener, Sonocytology and Cellular Sounds. This research is indeed fascinating and raises questions such as how are cellular vibrations converted into sounds that scientists can interpret as conveying meaningful information regarding the dynamism of cellular interiors? What conditions enable scientists to describe cells as actors capable of speaking or even screaming? What kinds of applications does this research foretold in terms of the medical field? The technology of Scientology, Roosth describes, produces an ambiguity between cells speaking and cells being spoken for. And as such, it demonstrates how scientists listening to cells transform raw sound into signal. After the talk, there will be a brief Q&A, so I hope you stay and ask some questions of the speaker. And now I'd like to offer a few words of introduction about Sophia. Sophia Roosth is currently assistant professor in the history of science at Harvard University. Her research focuses on the 20th and 21st century life sciences. Her first book, Manuscript, examines how the life sciences are changing at a moment when researchers build new biological systems in order to investigate how biology works. In this work, Roosth asks what happens to life as a conceptual category when experimentation and fabrication converge. Roosth joined the Department of the History of Science in 2011 at Harvard University. She previously worked as a postdoctoral fellow at the Pembroke Center for Teaching and Research on Women at Brown University, and she received her doctorate in science studies from MIT in 2010. Next year, she will be a fellow of the Radcliffe Institute for Advanced Study. Would you please all join me in welcoming Sophia Roosth?
Hello. Thanks to everyone for coming. I'd also like to thank the List for organizing this event. I'd like to thank Amalia Pica for inspiring this talk through her work. And I'd also like to thank Elizu Paitis for the introduction. How is the act of speech enabled by a listener? What gaps, stutters, and moments of miscommunication frustrate our efforts to enunciate ourselves, even as they make speech, and by extension, sociality, possible? How do we turn nonsense into sense, or sense into nonsense? The glossolalia Amalia Pica describes as babble, blabber, chatter, jibber-jabber, patter, prattle, rattle, yammer, yada, yada, yada. Pica's work tunes us in to breakdowns and errors in communication, the way information gets transmitted, though sometimes jammed and distorted by obsolete technologies. She reminds us that the listener always precedes a message's delivery. Not a word can be said without someone already available to lend an ear, even if the listener is mishearing. Now, while I'm aware that Pica is also experimenting with an audience who might be misunderstanding her own work, I'm going to take a risk in extending her interest in the listener, asking what happens when scientists listen to cells, exploring how they hear and sometimes mishear them. And if, in so doing, I myself am mishearing Pica's call, I hope that she's forgiving. because in so doing, I might also be inadvertently proving her point. The entity doing the speaking in my story is Saccharomyces cerevisiae, commonly known as yeast. Yeast is a unicellular fungus with a cell cycle similar to that of humans. The first eukaryote to have its genome fully sequenced, and a standard model organism in biology research, yeast is an organism that lends itself to multisensory encounters. It has been imaged extensively with light and atomic force microscopy, and anyone who's seen the bottom of a pint glass or has walked past a bakery can speak to yeast's olfactory and gustatory allures. It's fitting, then, that this species is also the first to have its cellular noises amplified and recorded. Sonocytology is a technique that was developed by nanoscientist and UCLA professor James Jimchevsky 10 years ago. It uses an atomic force microscope to record the vibrational movements of cell walls and then amplifies these vibrations so that humans can hear them. Yeast cells vibrate approximately 1,000 times per second, and humans can hear any vibration that has a periodicity in the range of 20 to 20,000 vibrations per second, or hertz. So the vibration of yeast cells is well within the frequency range of human hearing. In musical terms, it's about a C sharp to D above middle C. But the amplitude of their vibrations is too low to be within normal hearing range. So the cell wall of yeast is displaced only about 3 nanometers each time it vibrates. But by amplifying the vibrations of these cells, UCLA scientists are essentially turning up the volume on cellular vibrations. And what I want to ask in this talk is, how are these raw cellular vibrations converted into cellular sounds that scientists can interpret as conveying meaningful information regarding the dynamism of cellular interiors? What are the conditions that enable scientists to describe cells as actors capable of, to use their terms, speaking or screaming? And how might listening to cellular sounds eventually change how scientists think about cells? So today, I want to analyze how sonification constitutes scientific objects and how scientists, in listening to cells, represent these scientific objects as subjects that are capable of meaningful enunciation. The technique of sonocytology renders ambiguous the distinction between cells speaking and cells being spoken for. Specifically, I want to attend to how raw sound is transformed into signal. That is, how scientists can convert inchoate cellular vibrations into meaningful scientific data. And what I'm going to do is I'll begin with an overview of sonocytology, the technique, and describe how it was developed, how scientists and popular media have turned to metaphor in order to make use of cellular noises, or to make sense of cellular noises. And I'm then going to focus on the effects of using sound scientifically to explore otherwise inaccessible spaces. So specifically, the way we think of organisms in their environments and in relation to other organisms. By considering the diverse meanings of transduction, and I'll get more to what transduction means later, but for now I'm referring to the conversion of a signal from one medium to another. By considering these diverse meanings of transduction, I want to explore how sound travels through different material environments and how it is converted into scientific information. After addressing the influence of acoustics on the understanding of cellular milieus, I examine how scientists make sense of cellular noises, and more specifically, how sonification constructs a particular form of technologically and socially mediated cellular subjectivity. So Jim Jimchevsky is a distinguished professor in the Department of Chemistry and Biochemistry at UCLA, where he has taught since 2001. Jimchevsky is best known for nanotechnology research he conducted while at the IBM Zurich Research Laboratory. A celebrity in the nanotech world, he has received numerous honors and prizes, including the Feynman Prize in Nanotechnology. As a postdoctoral student and later a research team leader at IBM Zurich, he pioneered the use of scanning tunneling microscopes, what are now known as STMs. And this is the first scanning tunneling microscope that was developed in IBM Zurich. And just to give you a sense of how the scanning tunneling microscope works, there's a gap between the probe and the nanoscopic surface, and electrons actually move or tunnel between the probe and then the surface, and the current that's produced by those electrons tunneling in that space is directly related to the gap between the probe and the surface. So it can be converted into a three-dimensional topological map by measuring the current that's being transmitted. So Gerd Binnig and Heinrich Rohrer, who are the colleagues and supervisors of Jimczewski at IBM Zurich, received the Nobel Prize in 1986 for designing the STM. The microscope Jimczewski himself helped build at IBM is still in use in his UCLA laboratory, where I conducted anthropological fieldwork in 2009. Since its manufacture in the 1980s, the AFM, atomic force microscope, has become indispensable to nanotechnology work. While light microscopes cannot resolve objects smaller than half the length of a light wave, the AFM resolves this problem, which scientists term the Rayleigh limit, by using a probe to map the topology of the object being imaged. So the point here is that you can actually image objects that are smaller than half the length of a light wave by dispensing with light microscopy altogether. In 2004, Jimczewski intended to record the movement of cardiomyocytes, which are heart muscle cells that beat in culture. Atomic force microscopy had already been used for probing the surface of E. coli and imaging biomolecular reactions as they occur. The heart muscle cells were sent to him by Carlo Ventura, a Sardinian medical researcher he had met at a conference in 2001. Jimczewski's cells were scheduled to arrive from Sardinia on September 11th, 2001. But in the heightened state of national security on 9/11, the cells were deemed a potential threat and were seized by customs. Frustrated and impatient to begin his work with the AFM, Jimczewski borrowed a yeast culture from colleagues who were in a nearby UCLA lab down the hall. And when he put them under the AFM, he was surprised to discover that yeast vibrate with a regular periodicity. Though both AFM and STM are examples of a broader category of microscopy that nanotechnologists call scanning probe microscopy, the techniques by which each microscope interfaces with a sample are quite different. Unlike STM, which operates in this tunneling mode that I described earlier, in AFM, probes come much closer to sample surfaces. And this is a technique that researchers call contact mode. Members of Jimczewski's lab compare AFM reading the topology of a microscopic surface to a blind person running his finger over a line of Braille. Borrowing Thomas Kuhn's phrasing, Jimczewski called the shift from light microscopy to probe microscopy a major paradigm shift. Rather than using lenses and waves, they were recording by feeling. And just to explain the difference between scanning tunneling microscopy and atomic force microscopy, like I said, here the probe is coming much closer to the surface of the sample, and it's actually running in raster fashion over the sample. And what that means is it's deflected depending on the topography of the nanoscopic surface. As the tip is deflected, that mechanical motion is converted either by a laser or by what's called a piezoelectric crystal, and is converted into electrical current, which is then used to map the surface. So that's the difference between the two techniques. Most basically, the AFM is much, much closer to the sample surface. So as a tiny cantilever, and the tip is less than 10 nanometers wide, is displaced, this piezoelectric crystal converts the nanomechanical motion into voltage, which then creates this map of the surface. But the difference that Jimczewski made was instead of dragging the probe over the surface of the sample, he and his graduate student, whose name was Andrew Pelling, held the AFM probe stationary on the surface of the yeast cell so that the oscillations of the cell wall could themselves be traced. And this is the technique that Jimczewski named sonocytology, which he compares to using your finger to feel a pulse. Yeast cells are about 5 microns in length, and they have walls that are much more rigid than most mammalian cells, which makes it easier to rest a microscopic probe on their surfaces in order to detect cellular vibrations. So most eukaryotic cells are much softer than that. Jimczewski discovered that yeast cells vibrated rhythmically and that the periodicity of the vibration was within the range of human hearing. Using a freeware program he had downloaded from the internet, he converted the vibrations that he had recorded with the AFM into a sound file. And the resulting sound was something like this. That goes on for much longer. How do acoustic technologies change what it means for something to be audible, given that sound is by definition a vibration that can be heard by a listening organism? Jonathan Stern defines sound as a product of the human senses and not a thing in the world apart from humans. Extending Stern's definition to include listeners beyond the hominid, under the rubric of sound, I would include any vibration perceptible to any organism. A vibration is, of course, not necessarily audible, and sounds are not inherently meaningful. Only mechanical oscillations within a very small range of frequency and amplitude are audible without technical manipulation. So sound, then, is any vibration within the range of an organism's hearing, or, since the advent of acoustic technologies, of an organism acoustic machine assemblage. Listening, Pika reminds us, is conditioned by varied apparatuses that translate, transduce, encode, and decode messages. The artist signals our attention with semaphore flags, telegraph machines, TV antennae, and megaphones. Further, because sound is necessarily related to a biological sensorium and assumes a listener, it has a semiotic component that is parsed in historically, politically, and socially specific contexts. That is, listeners are the ones who get to decide what is sound and what is noise. If a signal is not deemed meaningful by a listener, then it is merely noise, irrelevant or superfluous information that can interfere with the transmission of information. A signal, in contrast, is a sound that a listener regards as conveying information about the source from which it comes. Apart from vibrations, which refer to a purely physical phenomenon in the world, sound, noise, signal, music, voice, and scream each assume a listener who can judge the ontology of an acoustic resonance. A listener designates a sound as music if he or she judges that someone composed it to be rhythmic, aesthetically pleasing, or otherwise expressive. Claiming that a sound is a voice imbues the sound source with the agency to utter sounds that convey information. A scream is inarticulate speech made by a human to express extreme pleasure or pain. Non-human animals are rarely described as screaming. They screech, they squeal, they yelp, or they howl. Attending to how cellular oscillations are alternately described as sound, noise, signals, music, singing, or speaking reveals the ways in which listeners interpret cellular agency and subjectivity. Parsing cellular signals from noise, then, is determined by researchers' understanding of cells as subjects capable of speaking to their own conditions. To sort out how sound affects the way we understand cellular interiors, I find it useful to employ Georges Canguilhem's use of milieu, and here is Canguilhem in his own milieu. And in defining milieu, he describes an array of decentered and mutually influential relations between an organism and its surrounding environment. Pica herself makes explicit how sounds and silence wash over two people attempting to communicate, most notably in Strangers and sorry for the metaphor number 2. Both pieces express distance, the social attempt to scale the intervals that separate us, and the shared milieus in which communication can occur. As Pica explains in conversation with João Ribas and Julie Widholm, the curators of the list and the MCA Chicago, for me, the literal physical distance from the source of the communicative attempt to the potential receiver is also the metaphorical distance. A lot of my work explores a face-to-face encounter, either through the analog technology they reference or through a direct encounter between an object and a viewer. By drawing listeners into the environment of its source, such communicative encounters produce a soundscape in which the different milieus of people and other people, or in my case, people and cells, can resonate. Beginning to experiment with the noise yeast produced when amplified, Jimczewski and Pelling recorded the vibrations yeast made at different temperatures and in different solutions. Adding sodium azide, a chemical that inhibits cellular metabolism, to the yeast caused a noise that sounded like radio static. That one also goes on for a long time. Jimczewski believes this sound is an indexical representation of the Brownian motion of molecules, since sodium azide stops all ATP-driven nanomechanical activity. That is, it shuts down cellular metabolism. On the other hand, when they doused the yeast in alcohol, the pitch of the vibration increased. In an interview, Jimczewski claimed that, and this is a quote, he said, "It screams, it doesn't like it. Of course, yeast produces alcohol, as in beer production, but if you put strong alcohol, like Absolute vodka on it, if you like, then it screams. It screams, it doesn't like it." He goes on to speculate that this screaming is the sound of molecular pumps working overtime to expel the alcohol from the cell. When he says that when doused with alcohol, yeast scream because they do not like it, Jimczewski endows yeast with agency. Characterizing the sounds made by yeast as screaming is an odd descriptive choice, as it suggests that Jimczewski's experimental interventions cause the yeast pain. Popular science articles about sonocytology picked up this metaphor, describing Jimczewski as, quote, the master of his cellular torture chamber. The suffering of model organisms, which makes most scientists quite uncomfortable, is usually expunged from professional and popular accounts of scientific research. Screaming is not just any kind of signal. It is an interrelational, emotionally loaded message, uttered either in pleasure or in pain. As cultural historian of sound Douglas Kahn puts it, screams demand urgent or empathetic responses and thereby create a concentrated social space bounded by their audibility. As a mode of communication, screaming is usually only attributed to humans, but here it is more than a response to environmental crisis. Interpreting cellular noise as screams forces attention on the shared cellularity of humans and yeast. Endowing yeast with agency by calling upon an anthropocentric model of subjectivity, scientists transform objects of scientific research into cellular subjects capable of speech. In her moon golem, Pika reinterprets a figurine astronaut David Scott left on the moon's surface in 1971. By projecting the letter H onto the icon, it is animated by the life of the mythic Judaic homunculus. As Anna Teixeira-Pinto interprets moon golem, an uncultured creature unable to speak, the golem is activated through language units. Golems, as Pika makes manifest, are at home in the information age, which sees the universe as essentially composed of signs and signals. So too, sonocytology animates and activates single-celled organisms with the breath of language, transducing their vibrations into signs that thereby encode their lively subjectivities. Sonocytology, and Jimchevsky's characterization of his technique in particular, is saturated with tropes borrowed from non-Judeo-Christian religions. And the notion of biological vibration has struck a chord with many representatives of such countercultural movements. So, for example, when Jimchevsky and Pelling published their findings in the journal Science, representatives of the Maharishi Mahesh Yogi approached them, thinking that they had found, as they put it, and this is a direct quote from the representatives of the yogi, discovered the language of life. The bookshelves of Jimchevsky's office are crowded with books that have been sent to him by people who read about sonocytology in the popular press and think that it might be one technique with which to scientifically validate their particular New Age philosophy. And here I'm including extrasensory perception, or ESP, but also Zen Buddhism, yoga, meditative auras, bioenergy, sound healing, biofeedback, and the list goes on. So for his part, Jimchevsky has invited Tibetan monks from the Gaden Lopa Kangsten monastery into his laboratory to meditate beside the scanning tunneling microscope and to compose sand mandalas. And in fact, on the day that they came to meditate beside the microscope, it was broken, and later in the day it started working again, and you can make of that whatever you will. Jimchevsky hopes that sonocytology, by registering biotic vibrations, will verify what he terms the subtle phenomena he considers to be central to multiple non-Euro-American medical, mystical, and spiritual traditions. Among them, he counts the principles of chi and chakra. More so, he believes that listening to biological vibrations will unite disparate theories of life, of life energies or life forces that are posited by such diverse traditions. Sound for him is not just an index of life, but also life's substrate. The sounds made by yeast also would provoke flights of metaphorical fancy, as scientists and journalists alike struggled to find words with which to describe something new in familiar terms. Popular press articles on sonocytology liken cell sounds to the whistling of singing cetaceans and compare the atomic force microscope to a microphone, a new musical instrument, and a record needle. Jimchevsky told reporters that if yeast were the size of humans, their sounds would be closer to the volume of ordinary conversation than of loud music, and that, quote, if you were to shrink down to the cell's size, it would be like holding a transistor radio to your ear. Lab members are also prone to thinking about their microscopes as audio technologies. At least one graduating PhD confided to me that he had independently made music using both nuclear magnetic resonance imaging and the decimal notation of pi. When a science journalist once described Jimchevsky as someone who and this is a quote from the science journalist, who might easily be mistaken for a refugee from an aging British rock band, she was not far off from the truth. In fact, before working at IBM Zurich in the 1970s, Jimczewski had indeed been a member of a British rock outfit that he had named Attention Deficit. So while graduate students would spend full days staring at computer screens, trying to make sense of data gleaned from AFM and STM scans, that is, as they would separate scientific sound from scientific noise, they also immersed themselves in very loud noises, most often in the form of industrial metal. When Jimczewski complained to me once that his students spent all day listening to that noise on his computer, I was unsure whether he was referring to his students' scientific data or his musical tastes. While the sounds produced by sonocytology are conversions of surface vibrations of yeast cells, researchers believe these sounds provide access to the workings of the cellular interior by signifying cellular metabolism and movement. So, describing the technique that he developed, Jimczewski says, "We gently touch a cell, a living cell, and we listen. They actually produce a kind of music, and you can hear it." He says the music made by the cell is amazing and beautiful. Now, characterizing a vibrating cell wall as music is predicated upon a definition of sound as something audible not only to the ear alone, but to the ear with the aid of technical amplification. In the 1960s, American composer John Cage traveled just down the street to Harvard to listen to his circulatory and nervous systems in an anechoic chamber. And this experience led him to conclude that the entire lived world is sonorous and resonant. As he put it, that we have no ears to hear the music the spores shot off from basidia make obliges us to busy ourselves microphonically. Yeast are already making music. We just have to heed John Cage's advice and busy ourselves microphonically. Jimczewski also compares listening to the vibrations of yeast to eavesdropping. He describes the experience as akin to standing outside of a factory and hearing the hum and beat of machines operating inside the factory walls. He points out that during the Industrial Revolution, trained mechanics could diagnose what was wrong with a machine just by listening to it. Extending and concretizing that analogy between cells and machines, Jimczewski is currently applying sonocytology to clinical diagnostics by listening for the difference between healthy and cancerous cells. He believes that sonocytology has potential diagnostic applications because cancerous cells metabolize more quickly and therefore vibrate at a higher frequency. And I've listened to some of these recordings. I'm no expert, but most people can hear the difference between a healthy cell and a cancerous cell. His ultimate goal is for clinicians to be able to detect cancer at an early stage just by listening to the cells. Now, cellular sounds are not meaningful to the cells themselves, but they could be made meaningful through human audition. Nonetheless, some scientists have suggested that the vibrations picked up by the AFM are indeed signals that the cells themselves are using to communicate with one another. So that is, they believe that the cells are already talking to one another, and we're just listening in. Carrie Bloom, who is a mycologist at the University of North Carolina, points out, and this is a quote again, that it was a big surprise when people played rock music to plants, and there was a chemical change inside the cell of the plant when you played the Stones at high volume. And so now I would argue the same thing with anything with a cell wall. Now the expectation is some physical output that can be another level of signaling. That's the end quote. So inspired by Pica's If These Walls Could Talk, I might retitle Sonocytology, and again with my sincere apologies to the artist, If These Cell Walls Could Talk. In a reverse game of telephone, garbled noises rewind into meaningful speech. Inscription devices turn occurrences into events, And in attempting to make sense of cellular noise, sonocytologists speak on the yeast's behalf. Historian of science Emily Thompson defines a soundscape as simultaneously a physical environment and a way of perceiving that environment. It is both a world and a culture constructed to make sense of that world. Bound up in the process of turning sound into data is the listener's culture, the environment in which the sound reverberates, and the material objects within that environment that create and sometimes destroy those sounds. That is, a listener is both acoustically and culturally immersed in soundscapes. But how does sound condition an organism's environment? And how does that environment affect which sounds count as signals and which are merely noise? Listening to cellular noises attunes scientists to cellular proxemics. And what I mean by that is the way that each cell is embedded in and in mutual relation to its microenvironment. Symmetrically, just as cellular noises draw attention to a cell's immersion in extracellular environments, and in this case I'm thinking of, for example, the very constructed environment of the laboratory, the interpretation of cellular noise is embedded in the listener's culture, so the culture of the scientist. By tying cellular and cultural immersion together, we can hear how listening to cells creates a space in which cellular and human milieus resonate. What then is the milieu of Jimchevsky's yeast cells? His atomic force microscope is housed in a special darkened, noise-free room inside a thermically, acoustically, and electrically isolated chamber lined with foil on a vibration-free platform suspended in air. The care taken in isolating the AFM from any vibration is necessary in order to verify that the vibrations recorded are in fact due to cellular activity and not to any kind of external noise. And here again, when I use the word noise, I mean both noise as an external phenomenon, so in the sense of sound, and also figuratively as a disturbance in scientific data. So my point here is that, ironically, in order to listen to the vibrations of cells, quote unquote, in their natural state, a very artificial environment must first be constructed. Listening to cells begins by demanding silence. As a relation between an organism and its ambient environment, a milieu is a landscape that influences and in turn is shaped by the organism that occupies it. The notion of milieu fastens organisms into the web of their environment's particularities, drawing attention to an organism's interaction with its environment and with the other organisms in it. In his explication of the milieu, French philosopher of science Georges Canguilhem writes that it explains the passage from the notion of fluid as a vehicle to its designation as a medium. The fluid is the intermediary between two bodies. It is their milieu, and to the extent that it penetrates these bodies, they are situated within it. He continues, circumstances and surroundings still retain a symbolic value, but milieu abandons any evocation other than a position indefinitely denied by exteriority. The now refers to the future, the here refers to its beyond, and so forth, always ad infinitum. The milieu is really a pure system of relationships without supports. Sound can draw attention to the milieu in which an organism is situated. Vibrations travel through air or water, and they refract off of other objects inhabiting the same milieu. So notice, for example, that all of the sounds Jimchevsky and Pelling recorded were differentiated by the type of environment in which the yeast cell was situated, which varied according to things like temperature, osmolarity, as well as the presence or absence of chemicals like sodium azide or ethanol. The recorded sounds as the index of cellular responses to extracellular circumstances demonstrate the porosity of the cell wall, right? That things can move between the inside and outside of the cell. And in so doing, blurs the boundary between intracellular and extracellular landscapes. So here it's worth noting that the experience of listening reconstitutes a parallel situation for the listener's bodily relation to his or her environment. So, for instance, sound studies scholar Julien Henrique describes the experience of listening to dub music as feeling the pressure of the weight of the air, like diving deep underwater, making the experience imminent, immediate, and unmediated. Sound is a system of relations between at least two bodies. It requires an origin as well as a receiver to sense audible vibrations. While sound has a point of origin, there is no center to the space through which it is transmitted. Bodies are both situated within an acoustic space and are penetrated by it. And as again, quoting Henrique, it is a kind of space you are inside as well as outside, and it is inside you as well as you being inside it. Marshall McLuhan speaks similarly of what he terms auditory space. It is the act of hearing that itself creates auditory space, because we hear from every direction at once. Auditory space, so crucial to architectural problems today, is usually defined as a field of simultaneous relations without center or periphery. That is, auditory space contains nothing and is contained in nothing. It is quite unvisualizable, and therefore, to the merely print-oriented man, it is unintelligible. Auditory space by necessity implies a listener who defines and demarcates it. So that is, by definition, auditory space must be a biological space, one inhabited by organisms that are busy making noise and listening to their own and each other's sounds. Neither immaterial nor purely informatic, as McLuhan might suggest, sound is a perceptual field requiring topographies, media of transmission, and listeners. A series of milieus is folded into the practice of sonocytology. Each milieu is in itself an array of relations that then becomes linked to other milieus. So to start, there are the milieus of the scientist, who might be ensconced in a vibration-free room, manipulating the probe of an STM or an AFM, or perhaps sitting in front of a computer, listening to the sounds of yeast flood from speakers into the lab. There is also the milieu of the yeast cell. Because the cell cannot be taken out of fluid without dying, it is suspended in a fluid yeast extract and flushed through a lattice of 5 micrometer pores, so that a yeast cell is trapped in each pore before being placed in a petri dish and then doused in yet another medium, which is made of pulverized potatoes. After being corralled and pinned by the tip of the AFM probe, the yeast vibrates in this isolated chamber. Beneath the cell wall lies a cytoplasmic milieu, inhabited by organelles suspended in cytoplasm, and motor proteins that transduce chemical energy from ATP into motor energy in order to build cellular scaffolding and to traffic molecules through the cell. The transduction of sound from each of these milieus to the next constructs a soundscape where cellular processes become audible to biologists, or at least it becomes audible to the biologists once they have learned how to listen to what it is they are hearing. Resonances scale the domains of previously isolated milieus. An acoustic milieu, then, is one shared by 2 or more organisms in relation to each other and to their surroundings. So if, as Canguilhem writes, the milieu that is proper to man is the world of his perception, then listening to yeast creates a space that the scientist shares with yeast. It is within this thumping cytoplasmic milieu that we can imagine ourselves when listening to cellular sound. But listening occurs in time, and cellular activity is dynamic. So we must also attend to the modes in which sound is transmitted through acoustic milieus. Sound is transduced as it travels through media and mediating machines. Transduction, as engineers use the term, refers to the technically mediated process by which mechanical vibrations are converted into electrical signals. Emily Thompson argues, the historian of sound that I mentioned earlier, that the technical and material development of transducers in the 1920s and 1930s significantly affected the epistemology of sound. As she explains, scientists who used electroacoustic transducers began to effect similar transformations between sounds and signals in their minds. They developed new ideas about the behavior of sound and the physical objects that produced it. So another way of putting this is that by turning sound into an electrical signal that could be amplified, manipulated, transformed, etc., acoustic technology turned sound into information that could be fruitfully studied by scientists and then used as data for gathering information about natural phenomena. Transduction has 3 definitions, all of which apply to sonocytology. Acoustic transduction is the conversion of a signal, such as a sound wave, from one medium to another. Biological transduction is the transfer of biological information from one organism to another, or the translation of a stimulus into an electrical impulse. Third, technical transduction converts input energy into output energy of a different form by a transducer. And among transducers, you could include a microphone or the piezoelectric probe of an atomic force microscope. Piezoelectricity defines this reversible conversion of mechanical energy into electricity. So, for instance, microphones transduce mechanical vibrations into electrical signals, while speakers do the reverse. In addition to microphones and speakers, I should note that a third kind of transducer is also at work here. Namely, the human sensorium can be understood as a device for converting mechanical energy, light, and chemical stimuli into electrical impulses, as when, for example, when I was doing fieldwork in the laboratory, a postdoc told me that piezoelectric crystals work just like my nervous system. The yeast AFM human assemblage that performs sonocytology is a series of vibrations traveling through different media and converted into sound by mediating transducers. The kinetic motion of motor proteins becomes a cytoplasmic rumble that vibrates the cell wall, which exerts pressure on a cantilever, causing the piezoelectric crystal to convert the deflection into an electrical output. A graphic trace of its deflection is created, which is then converted using a computer program into an electrical signal, which is then sent through a pair of speakers as a mechanical wave oscillation. This creates a periodic turbulence in the air that vibrates the eardrum, which vibrates the ossicles, which vibrates the fluid of the cochlea, which ultimately triggers hair cells to send electrical signals to nerves that then travel to the brain. Each time the signal travels from one neuron to another, it must be transduced from electrical to chemical energy while traveling through the intercellular synapse, that is the gap between two nerves. Thus, the acoustic, the technological, and the biological harmonize with one another in a biological soundscape. However, this biological soundscape is in turn culturally transduced, obscuring the technical conditions of its production. Pressing our ears to opaque cell walls, we listen to cellular activity. The busy hum of actin and myosin filaments assembling cellular scaffolding, the whoosh of molecular transport through cytosol, the glub-glub of endocytosis and exocytosis. Listening to the soothing hum and thump of yeast metabolism allows one to imaginatively project a listening body into a yeast cell. Sound maps the dimensions and characteristics of the acoustic space through which it is propagated. Sound waves originating in one place extend outwards in concentric circles, slowing their pace through liquid media, diffracting or reflecting off of walls or other solid objects. Such qualities of sound, for instance, are utilized in sonar to orient objects underwater. Sonocytology, in turn, orients listeners to intracellular activity, cluing listeners in to the dynamism on the other side of the cell wall. Sound has been used in science to explore and gain direct experience of inaccessible places, to sound the depth of the ocean, for example, or the inside of a body, the furthest reaches of space. Acoustic technology is also used to connect with absent loved ones, as when telephone wires and satellites transmit disembodied voices, or with people on the margins of life, as in the early use of sound recording technology to embalm the voices of the dying, and the much more recent use of ultrasound in obstetrics. Scientists are increasingly becoming listeners. They're auditing the utterances of the natural world as raw scientific data. And examples of this impulse include listening to whale song, but also the reverb from the Big Bang, the groans of melting icebergs, the growls of active volcanoes, the rasping creak of a slime mold as it extends protoplasmic tubes outward in search of food. Yet when sonocytologists say that a cell is speaking, they're projecting cultural notions of what it means to be human, to be subjective, to be agential, even to be meaningful onto cells. So perhaps then, sonocytology is a mode of imperialism, seizing upon a cellular colony and asking that its epistemology resonate with our own, asking further that it communicate seamlessly and intelligibly, even as the technologies rendering it audible are obscured by the sheer persuasiveness of the cell's arresting and quite fluent volubility. This possibility reminds us, as does Amalia Pica, of the limits of listening. To listen to something is always to speak for it. Thank you. So I think I can take questions if anyone has any. Yes.
So presumably these vibrations, that, as you were saying, are not limited only to cells, for example.
Mm-hmm.
The sound, it's not just at that level, that scale of organization that sounds are emitted from. It's just the lowest level that the STM can go. Presumably anything that vibrates, like Brownian motion, like you're saying, would have a similar sound. If we could get that close, if we get closer to it, we could somehow model it or transmit it or transduce it. Is that true? I mean, so that there's sounds all the way down.
Right, so if I understand your question, you're saying that by amplifying vibrations, that means that any vibration could be understood as sound. And I mean, that's absolutely right. The difference with atomic force microscopy and its use in sonocytology, at least for the scientists who are doing the research, is that while the amplitude is much too small to be within the range of human hearing, the frequency is within the range of human hearing. And so they consider that to be significant. But that being said, one of the points I was trying to make was that indeed, when you start talking about amplifying certain kinds of vibrations, recording them and then turning them into something that can be heard by humans, that does extend outward to include many different kinds of vibrations and actually problematizes how we think about the relationship between what might count as a vibration, what's noise, and what is sound. Sound becomes a category that is itself up for debate. And the examples of scientists who have been listening to new kinds of sounds in recent years, I listed things like volcanoes and the sounds that are made by slime molds as they move their protoplasmic tubes outward. All of those are also vibrations, and they're heavily technologically mediated. And these are examples of what's called infrasound, so something that's sub-audible and yet amplified by scientists, and something that I'm hoping to pursue in a later project. But the point here is that by trying to think about that as sound, any kind of vibration of the natural world, like you said, becomes thought of not just as something audible, but as something meaningful by dint of its audition. Yes?
It seems to be there's two categories of things that produce sound. One would be something like a volcano or a storm or something of this sort, which we consider to not be a living entity, although it may be, some kind of an automata of interacting cells, even though they may be transitory, and something which we consider a living organism, be it an insect, a bird, a human, or what have you. And we clearly know that there is a communication going on between living forms, insects, birds, and so on, and that, in fact, they pick up these cues, they use them for predatory and prey, and so on, type of reactions. So my question is, in this whole area of sonocytology, has there been any study of the communication between the lowest level of living forms, which would be the cells, and if in fact one sees that the formation, for example, of bacteria colonies and so on, that there was some kind of a communication going on, much like birds communicate or insects communicate with sound, and it helps them to socially and otherwise organize. Has anyone seen anything? Has the sound been looked at in terms of larger colonies?
It's an interesting question. So you're asking whether, in fact, these vibrations are a form of cellular signaling.
Yeah.
And there's been a lot of speculation about that. There haven't been any studies that are done, but that's what I was pointing to in that quote by Carrie Bloom, the mycologist. So some scientists do think that perhaps these kinds of vibrations might be a form of intercellular signaling, the ways in which cells communicate with one another to do things like form colonies, but there's absolutely no scientific evidence that that's the case. And the fact that the cells vibrate differently depending on the environmental stimulus or pressure that's placed on them suggests that perhaps it really is just an index of things like metabolism rather than some kind of signaling effect.
What I was really asking also was that given that your talk, how do you stand on this? Are you seeing humans as overly humanizing, or as you said, imperialistically imposing, or do you, on one hand, which is purely of a human psychological nature, or do you feel that at a scientific nature there is a whole reality, as I suggested by the first question, going on?
So the question is whether I think about the scientists as overly interpreting the sounds?
How much of it is that, and how much, but beyond that, is there a real science going on there?
Oh, I have no doubt that there's real science going on. I've been in the lab, and they're definitely doing real science and producing results. But I should say that the fact that there's real science going on doesn't mean that there isn't also lots of cultural work going on. And what I'm trying to point to here is that there is something fundamentally different in listening to cells than in looking at them under a microscope. And when we start talking about sound, then there are all sorts of ideas about how we think about sound and what it means to be able to hear something, which are culturally situated and historically specific. That changes the way we then think about cells. Specifically, what I'm pointing to here is the subjectivity of cells, but also, and I go into this in more detail elsewhere, the dynamism of cells, the way we think about cells as temporal entities that unfold in 4 dimensions. And so the difference between visualizing something living and listening to it is cultural work that gets done in a scientific context.
Because, you know, during the past centuries, scientists are always, you know, very eager observers, right? So, I mean, and you just mentioned that scientists has already become a listeners. So I'm wondering how can you put, you know, your story into the history of scientific observation or, you know, objectivity?
So the question is how I would place this particular case within the history of science. And I have an answer about the history of science, and I have an answer about the historiography of science. So in the history of science, I think that, like I said, scientists are increasingly becoming listeners. When I say that, I don't mean that scientists weren't listening before. They certainly were. But I think that sound is increasingly becoming a form of data. So it's an object of scientific inquiry rather than just a form of scientific research or a method. But there are plenty of earlier examples. I mean, even in the mid-20th century, if you look at oceanography, heavily influenced by the use of sonar, and scientists were trained to listen. Within history of medicine, you can look at the way scientists were trained to be able to use mediate auscultation as a diagnostic tool. And those are examples not just of listening, but learning how to become good at listening for certain kinds of information. So that, I think, would place it within the history of science. But one of the interventions I'm trying to do here has less to do with the history of science and more to do with the historiography of science. And we're so influenced in the historiography of science by the very good literature that's been done on scientific visualization, the way we've looked at graphs and charts and maps. In so doing, you can think of the history of microscopes and telescopes being part of that historiography as well. I think there's been less attention in the last few decades to other forms of scientific inquiry that involve senses other than the visual. And here I include hearing, but also smell, taste, touch, as very valuable forms of scientific research, both within the laboratory, but also in the clinic, in the field, and beyond. Yes?
How do you expect it to, the field sonocytology, is that right?
Sonocytology, yes.
Sorry. I'm word jargon. But I was wondering about how do you see the opportunities to expand language and like grammar and how we think about, like, is sound enough of a word for it? We already have good, like, sound is a good specific one because we already have like a differentiation for like air and wind and sound, like the air itself, the waves of the stuff, and then the things propagated through the medium.
Mm-hmm.
There's things like that make electricity hard to understand because we use like one word for it, and we don't think about the electrons and like the wave of the electron flow versus the like propagation of energy that flows through the column of electrons. Like there's kind of like sound, wind, air corollaries there that like how do you think that our language is weak in any spots that we don't have enough differentiation in the way we use words for these things, or do you think there's ways to integrate across fields, like in how we phrase these things that are like I don't know, that move? How do we go beyond the agency-based things? I don't know. This is an open space. So yeah. Did you catch?
I'm not quite sure I understand the question. Are you looking for an expanded vocabulary with which to talk about?
Yeah, does sound encompass the space, or does milieu and like I'm sorry, I can't.
No, it's fine.
My milieu is too like too embedded. Yeah.
So, I mean, if I understand your question correctly, you're asking whether there are other ways to talk about what scientists are hearing beyond sound, and that specifically sound can assume ideas about agency. Is that?
Yeah, and well, I guess in the process of like, is our agency being projected through the way the words we choose to interpret them as? Like,
Certainly, always.
Yeah, are we not differentiating enough somewhere, and that's where the agency problem is sneaking in, or is that not a problem? Maybe it's like,
Well, I don't think the agency problem is a problem at all, and I'm not trying to make a normative claim here, and I'm not suggesting that sonocytologists do anything differently or talk about what they're doing differently. As an anthropologist, I'm really interested in the way people talk about what they're doing, but I don't think that there's any kind of disconnect or miscommunication going on when sonocytologists choose to describe their cells as subjects. That being said, absolutely, the way we describe something about the world is going to influence the way we think about it. The fact that people are talking about the cells as screaming is going to change the way people think about cells, specifically as subjects and as agents. And yet I don't mean to suggest in any way that that should be done differently. I think, if anything, what I'm trying to do is more diagnostic and try to point in new directions as far as how the use of sound will change the way people think about cells without trying to say anything about whether that's the right or wrong way to think about cells.
do you think that'll be through new language, new stories coming out, or new ways to envision it, or like what do you
The ideal byproduct of sonocytology.
In order to convey these things, in the end, like, is it new ways to describe the landscape, or sorry, yeah, we should just talk later, maybe.
I'd be happy to talk later. I think less new, I would say that while shying away from the word ideal, I think different ways of thinking about cells is certainly in the offing. Well, thank you so much.